Yeah, he said that. हाँ So good morning, students. Am I
Good
audible?
afternoon, sir.
आज आज कोई नहीं आया. आज
No,
सब भाग गए. हाँ? ये गलत चीज है वैसे. Now it's your is online, offline, so it should be offline. And from Monday onward, there is going to be no excuse. There is be no online class from Monday onward. Okay. So आज तो चलो आपका दिवाली का break. So for that you can go. We can conduct online classes. But otherwise, from 8th onward, you have to be in the campus. However, it is just 1 week before your exam for ending your session, but it is going to be offline only. Okay, so we have to complete our last module, and last module is about gene cloning. And what this gene cloning is, so as we discussing from the first day itself, uh, gene cloning simply means that you are going to getting multiple copy of the same gene. So, but if fast as we are talking about history, so what is history of this gene cloning is going to be? Because this history of the gene cloning is when we talk about the gene, it is going to be associated with the genetics. And as your genetics is to keep on developing our molecular biology techniques, but we are going to use nowadays, because nowadays we are really refer the genetics, because nowadays we are become a bit more sophisticated, and we start talking about gene, how they are going to go further. So basically, your tech the modern days is going to be, it's not very old. The technique that we are going to use these days are just after discovery of your PCR, they become more advanced. Or if you talk about after discovery of your restriction enzymes, they become more advanced. But before that, we were dependent upon your old genetics, like your process, etc. Those type of things. So here the just your timeline, what will happen. So now, as I mentioned, the polymerase PCR, the Calimorellis gave, and restriction to nuclease uh, about your McCarthy and McLeod. So all these things. So now we need to reach to today's level, that now we are days we are talking about cloning, and not just the cloning, also about the recombinant DNA technology, the name of your subject also. And in this technology, we are going to deal about whatever the red highlighted things are you going to see here. This simple thing: gene cloning, DNA sequencing, determination of structure, genome sequencing, not just DNA sequencing, whole genome sequencing. And after that, we are going to talk about the recombinant one, and gene regulation, expression of your recombinant things. So all these things we are doing nowadays. And nowadays, whatever we are talking about the modern biotechnology, all our modern biotechnology is based upon this molecule cloning or gene cloning, and because we are talking about your modified or genetically modified organisms, GMO crops, etc. So this is a Carimales who described in 1985, and that leads to that make us easier getting multiple copy of a gene. So multiple of copy of a gene, how we are obtaining? One way is the PCR, by which you can obtain the multiple copy. But bring it to the functional way, we need to go for your host, so that you need to combine a recombinant one. So lots of application of PCR is there in the molecular ecology, forensic. So all these are there because of this PCR itself. So now is the main thing, that the brief history that we talked till now, and this brief history is just about what we have discussed in our all four modules. So they are the how we reached to till now. That is your four modules are there. We talked about your enzymes. We talk about your vectors. We talk about DNA sequencing. We talk about the identification, like the hybridization technique, and so on. So, when you're talking about gene cloning, it simply means you are making multiple copy of a gene. But when we are talking about multiple copy, it means where you have to produce your multiple copy. Simple thing is you are going to construct a recombinant DNA molecule. And recombinant DNA molecule you are making where? We are making that in a vector. So, it means we are going to have a vector. In that vector, you have to insert your DNA fragment. And when we talk about the inserting DNA fragment, and when you insert it, that you have got DNA recombinant DNA molecules. So how you obtain it? So it simply means you are going to cut it your vector so that it's opened up, and after opening it up, so you have to utilize some enzymes that are your restriction enzymes. Then you have to add your fragment, and you have to ligate it, so that you need a ligase enzyme, and then ligase enzyme join it up, and then it leads to the formation of your recombinant DNA molecule, that you need to transform it to bacterium, and your bacterium is that you call the host cell. So host is going to maintain this recombinant plasmid, and this host, to this host, which is now you are calling it a transformed host. So this transformed host, we are calling it as a clone.
So this transformed host containing one type of recombinant DNA molecule, we are using term clone for that. Okay. So when you're calling a recombinant clone, that means a cell, a host cell that contains recombinant DNA molecule, or it should contain only one type of DNA molecule. Then only it is going to be clone. Otherwise, it is going to be a mixture. Okay. So as we are talking about the steps, so these are just simple step how we have to proceed for the gene cloning. So gene cloning simply whenever the term gene cloning means you have to generate a copy. So okay, so within a cell, a host is your vector is going to multiply. This is a simple, nothing new. We already have talked about these things in your when we talk about vector itself. So here also the things are proceeding further. Now you have transformed your vector into a host cell. Now within the host cell, what they're going to have, your vector is going to multiply. It's going to generate an identical copy of itself, and that gene it carry. So that depends upon the copy number, how many copies it is formed. That is the property of your vector itself. So when a host divide, then it is going to pass these into a progeny of cell. That is a vector application take place. So now the vector is present in 1. Now it is into 2. So this is the how this cells are going to proliferating. When they are proliferating, then you are going to get a colony of cells, colony of a single cell. To that we call the clone. So after a large number of cell division, a colony or a clone or of an identical host cell is being produced. So this is the one. This you have to refer, but to whom we are calling a clone. So clone means identical host cell containing a recombinant molecule. So okay. So depending on how many recombinant molecules are there, how many type of clones are there, we are going to refer them as a library later on. Okay, so your cell division resulting into a clone. So this is a clone. Okay, so these are types of when you transform them, when you grow into a bacterial on a plate, then you are going to get a cell, a colony. So this is you are going to see in your plate. So each cell in a clone contains one or more copies of recombinant DNA. So here we are talking about copies. Here we are not talking about two different, one of a recombinant of the recombinant DNA molecule. Okay, so a gene carried by recombinant DNA molecule is now said to be cloned. So now this who is cloned? Here the gene is cloned. So term that we are using in the gene cloning is a gene. We are cloning the gene. So these cells, the bacterial cell, the host cell that we are referring, they are themselves cloned because they are replicating sexual replication. So getting a same copy of same type of cells. But what we are referring here, we have to refer a recombinant DNA. But we are talking about the clone of a recombinant DNA. It's your cell that we are saying, the host cell, which is there. This is a clone which they contain. What do they contain? They contain a gene that we are having. The gene which we transfer to a recombinant vector, that will call the recombinant DNA molecule. That recombinant DNA molecule will transform to a cell. It will grow. The colony is going to be produced. That colony is said to be a clone of a gene. Okay, gene carried by recombinant molecule is said to be now clone. Now this is clone because every time just grow these cells, you are growing the recombinant molecule, recombinant gene also. So that's why we are referring to clone gene. Okay. So now another type of thing is the method of cloning that we use the PCR. So what is the PCR? That we already discussed about this polymerase chain reaction. This is another way of a cloning, but this is a different, very different from the gene cloning that we are referring in case of your cellular cell host based therapy. So how it is going to differ? It is simply a series of because in case of your gene cloning method, when you are generating a multiple copy of a gene, many steps you are being involved. You are talking about your host cell, you are talking about your vector, recombinant vector. So many things are there. So you have to transform, you have to make competent, you have to ligate, so lots of things you have to do there. But in PCR, just you can generate a multiple copy of the same gene just simply in a simple way. So it is a bit simple rather than the manipulation that you need in living cells. As you need a marker there, you need lots of things. But you can do here PCR just get on a single test tube. This is the beauty of PCR. This is also a cloning method, so you can generate a multiple clone of it. So piece of that you need. So this is the background one. So PCR, how it is working. What PCR is? PCR is simply a thermocycler that can generate cycles of thermal different temperature. So in which your DNA is being heated. That we already discussed about how this PCR is working. This when you heated it, that's play role in the denature of your template. Your template become single-stranded. Your hydrogen bonds that were there, they got broken down. Now the double-stranded changes to the single-stranded one. This step is called the denaturation. That okay. So then you mixed cool it down. So when you cool it down to 50-60 degrees Celsius, what is doing? It allow the formation of hydrogen bond. What type of hydrogen bond is going to form? Okay. So this is the way that we call it the annealing. So what happened when you bring temperature down? So it's not happened that all the your normal molecules were formed. So you introduce one more thing. That is a short DNA molecule. That short DNA molecule will be polymerized as a primer. So now this primer is going to bind to your DNA molecule depending on that we are talking about temperature of annealing. So now these molecules they bind because they are present in the large excess. So one more thing is that if the primer amount is going to be less, then self-annealing is going to be there. Means your DNA, double DNA strand that we are talking about, that will self-anneal. When it self-anneal, then there is not going to be any amplification. Okay, so that's the oligo primer that you are going to add. So this primer will bind on the basis of complementary at specific position. So as you can see, this binds your the primer. So now the job of DNA polymerase is going to come. Now you have again to rise the temperature. You rise the temperature to 74 degrees Celsius. Now what is going to happen? This is a temperature at which your DNA polymerase, thermostable DNA polymerase, utilizing it there, that is going to amplify your molecule. So this is the simple thing. Now it is done, completes when it is over. So you have amplified the target gene. So amplified target means you have generated a multiple copy of a target gene. Okay, so now four strand of DNA molecules are there, two of each one to start. So when you start with the two, now you are going to have four. So now you can repeat the cycles. When you repeat the cycle, then we call it thermocyclers. So that is cyclic way it is repeating. So after again this extension, when the temperature is 74, you again go down to the 94, then to the 50 to 60, then again to the okay. But one thing you note that this condition that you are keeping it, we may referred in your PCR when you're taking the lecture of PCR, we mentioned that every condition, your time and temperature all depends upon the what set of primers you are going to have, how much length you are going to amplify. That is going to determine how long you have to run your polymerase chain reaction. Okay, so this is the way the temperature again you need to be increased. When you increase the temperature, then denature. So just a minute. Uh, okay, students, so I'm back. So this is a cyclic way when you're performing, and increase the temperature. So if you continue the cycle, each at every time it is going to be doubled up. So starting the two strand, if you repeating the 30 cycles, and it is going to be okay. Sorry, network got disconnected.
Okay, so this is the how and what number of the fragment you are going depending on the number of cycles. So this all we have talked about in your PCR itself. This is the way of your gene cloning. You are amplifying it. So now we talk about the why they are so important. They are so important because you want to obtain pure gene. So you can obtain both by both the methods. You can obtain the pure gene by the cloning way, that you are placing a cellular host, or a PCR. You can get a pure gene because PCR also amplifying the segment that you need. In the clone also, you are going to transfer the gene of your interest to them. You can purify the gene. So these are the way why it is so important. You can obtain the gene and you can purify the gene by both the way, gene cloning and the PCR. So now the question is, now we have to perform the your cloning experiment. So now for the cloning, how to obtain a pure sample of a gene. So for by the cloning, because the main aim that I will mention that both whether you're going for PCR or you're going for the gene cloning, your main aim is to get a pure gene. Just we are just referring the gene. So for how you are going to get that gene, that is the main the thing that we are going to deal with. So DNA fragment that we are cloning. So the gene that you need, that is going to be interest. So where that is present, that first you need to know. We all know that the gene is present on your chromosomes. So then the next question is, we know the location, but we should know that how that the DNA the genes are going to be different. We know that the genes are different on the basis of their sequence. Now, two genes are different between their sequence. So now we need to focus on the gene that we are talking about. It means the sequence that we referred up. So DNA fragment to be cloned is one member of a mixture of many different fragments, each carrying different gene or part of gene. So this is the one that we're talking about. So that the cloning is a part of a whole genome that you are referring. So from that genome you need to take it out. So for that, what you need to do, we need to perform. We need to generate a fragment. So means every fragment is there. It's not just a one fragment. You are going to have a large number of fragments. So these fragments are going to be kept. So just isolate when you are going to isolate your genome, isolate your genetic material, then break it with the restriction and nuclease. Then you are going to have a large number of fragments. So all these fragments you have to clone. Okay. So this is the one. Each of these fragments is inserted into a different vector. So these every part when you are taking fragment, when you take a mixture of the fragment in the gene cloning, so every fragment is going to be the going to join. It is going to join randomly. We can't say that which is only going to join. Everyone is have to join, but as a random event. Some is going to join more frequently, others are going to be less frequently, depending on their size what you are going to have. Okay, so a molecule produce a family of recombinant DNA molecules. So what you are going to get here, you are going to get a family, one of which carries gene of interest. So this is a large number, this family. This family means that contain the recombinant fragments. And out of this large number of recombinant fragments, only one is going to vector or one clone is going to contain your gene of interest. So each one is going to have, you can see in the side image also itself. So they are present. So only one recombinant DNA molecule transported into a single cell host. So out of three are there. So cell host is going to carry only one, not all. So now which one you need? When you are plating it out, so all of one are going to produce an individual colonies, and each colony is going to represent an individual clone. So now it's the job on us is we need to check which clone contain what fragment. So final set clone may be contain many different recombinants. So there are the different fragments are there in each cell. Individual clones contain multiple copies of just one molecule. But large number of clones are there in it. So it's a mixture. So you need to screen, you need to separate that which clone contain your gene of interest. So gene now separated from all other genes in the original mixture, and its specific feature can be studied. So now the separate you need to obtain it, you need to separate it. Success or failure of gene cloning is the ability to identify the particular clone. So now, as mentioned, you have to cloned it, you have transformed it, but this is a mixture you have obtained from a mixture of genes like genome and 50, 60 fragments. So every fragment is going to be transformed
So you have to identify. So if you're not identifying a right gene, a right clone, then your job is not done. So that means the main aim is the identification. So here's a simple example. If you can see, E. coli genome contains 4,000 different genes. So there are so large number of genes are there. So when you are cloning it, so it is difficult to find one gene among all possible clones. So you are going to have, if you say that the 4,000 genes are there, and these 4,000 genes you have cloned in different clones, different recombinant zones, then which one out of 4,000 is going to contain your swans? Then it is going to be difficult. Now that's the main thing, how to obtain that pure gene. So but if we go to further the complex molecule, complex genome, the complex genome like human or higher eukaryotes, like if you take an example of E. coli, just 4,000 genes are there. But if we talk about human, we are having a 50,000 around 50,000 genes. So when genes are so large, even 10 times than the gene size of your E. coli, then how we are going to handle so large? So then the variety of different strategies that we need to use to identify correct gene. And when you can identify the correct gene, you can isolate it or you can separate it from the mixture or the family that we are referring. Okay, so this is the thing that we have to refer. So what you need to do? So you need to change your things, involve modification to basic cloning procedure. So how you can scrutinize it, how you can reach to the gene of interest. So you can modify something so that you can decrease the number of your clone so that it's been easy to for the identification. Only cells containing desired recombinant molecule can divide and other clones just automatically selected. This is the one thing that we can say. So if only the cell containing desired recombinant DNA molecule. Now if certain things are associated with desired DNA molecule, if you allow that only to divide and others not divide, so by that way you can do nothing, but you can say how you can go about that. That involve enable desired clone and a mixture of lots of different clones. Now I think that first one we can't do, then a second one thing, a large mixture is there. So in that mixture we have something that make us to separate the desired one that we don't employ, like the method that we talked about, the hybridization that we are referring, southern hybridization that we are referring. So these technology techniques we can utilize here that you to know which gene is going to be of interest that you have cloned. So that will help you in the isolating that gene. So like a simple example, you can see that the gene to be cloned, gene what you want that tryptophan A gene to be cloned, like this whole genome is there. So your aim is to isolate this gene. So this is the protecting the process for you to clone that you need to check. So here you have information about it, that tryptophan A gene is there. It is going to be involved in the tryptophan synthesis. So one gene is one gene has been cloned. There is almost no limit to information that can be obtained out the structure and expansion. So this is the once you clone that you know the gene and you can identify, you can work over it, but still it's not there, then you can't. So here you can see. So when you digest it, so a large number of fragments you are going to get, like whatever the fragments you are going to have, everything is going to be fragmented. But your interest is with A. So now your aim is to identify it. So the availability of clone metal has stimulated the development of analytical methods studying gene with new techniques being introduced all the time. So this is the main aim and the main emphasis here is to how to isolate the gene that you need. So this is the only what you want to select. We want to select a recombinant which contain the trip A. So this is that is the main aim. So how you are going to obtain, how you are going to select this gene when you are going to transform. So PCR is a simple method that you can see to purify the gene, because in this case you need not to go for anything. But in previous one thing is that if you know not having any information about the tryptophan A, you can go with, you can select, you can clone it. But in this one, PCR one, if you have some prior information about your gene, then you can apply. Then you can design a primer and you can amplify it. So like here you are having a tryptophan A gene and you want to amplify it. Just a moment.
Okay, so it's another way. So to clone the gene, that is the PCR that we are referring. So region starting the DNA molecule is copied, so that is going to tell you the boundaries. So these boundaries, on the basis you can design your primers that are going to be your annealing position. You can design a primer for this that we already discussed in case of when you are talking about the primer designing. These primers are going to anneal on this boundary and they are going to amplify only gene that you are interested. So any copy of gene is going to be optimal. This simple way. Now you are having the gene, you can go with it. So what you are going to have in this way, you are going to have only the copy of your gene of interest. So these are two different ways by we can go. But here is a benefit that we are making in previous one that we are talking about the problem selection. Here the problem selection is not there because here the amplification that we are talking about, this amplification is based automatically based upon the boundary that you have already set. So whatever gene is going to be amplified here, it is going to be your gene of interest. So we need not to bother about the screening process here. Just what you are getting, if amplification is going to be there, that is going to be. But to check here by just amplification, you can't say that your amplification is going to be well good. But for that, you need to go for the genome sequence. You just get the sequence, and you will have uh, the thing. So, but now a question is that you may ask that, sir, why PCR is so simple and you can amplify a gene, but still we believe in the gene cloning, still you need a host cell. So simple reason is the host cell is here you can functional study of the gene that you want to do. That is easy to do in a cloning. So this is a simple thing. PCR experiment can be complete few hours, where it will take in the weeks to obtain gene by cloning. PCR is have two limitations. One limitation, in order to primer anneal the correct position, you must be aware about the genome sequence. Otherwise, it may, if it anneal at a wrong location, then it is going to amplify a wrong gene. So, thing here is that you must be aware about the sequence of gene, or you must be aware about the annealing site. Otherwise, if annealing site are not known, then you can't use PCR for amplifying your gene. Second limitation is it is easy to synthesize a primer with a predetermined sequence. Sequence of annealing sites are unknown, then appropriate primers cannot be made. This is the one thing that we do. And next thing is that second one, PCR cannot be used to isolate the gene that do not have been studied before. So if you are talking about a new gene, you are not aware about the gene, that you can't do using the PCR. So this is a limitation. This is the limitation of your PCR, that you can't clone a gene about whom you do not know or which is not previously studied. So this is the one that you're talking about. So there is a limit of length also. PCR cannot amplify a very large sequence. So limitation is a 5 kilobase pair. So 5, if you want to amplify a gene, if you want to clone a gene, multiply a gene by using PCR, 5 KB is fairly good. But if you want to work more than 5, like segment like 40 KB, if you want to work with, then PCR is not a good thing for you. And for that, you need to go for your normal cloning methods, or that you call gene cloning, like just a stick digest, get fragments, and clone. But this is shorter than the length of many genes. Okay, usually those for human and other vertebrates, so they contain very large genes. Cloning must be used if an intact version of long gene is required. Okay, so this is things. Okay, so now our time is also rushing up. So please now ending this session. So please turn on your camera, and then you may leave the team for recording your attendance. Adish, Atoyba, Devanshi, all of you turn on your camera. Turn on your camera. Those who have turned on their camera, they may leave the team also.
Thank you, sir.
Welcome. And happy Diwali to all.
Sir.
Happy Diwali, sir.
Happy Diwali, sir.